We're going to discuss about the topic biotechnology, principles and processes. What is biotechnology? As you can see, there is two terms, bio and technology. That means usage of live organisms. We'll see the definition. Here, you can see biotechnology deals with technique of using live organisms or its enzyme to make product and processes useful for humans or human welfare. So it is a technique where we use the live organism for making products for us. The term biotechnology was first introduced by Carl Ehrlich. Okay, it was first introduced by Carl Ehrlich in 1919. He was an Hungarian scientist, okay, where this technique was introduced. This term was introduced. Now, there is a lot of organizations in biotechnology where a lot of definitions is available. For example, European Federation of Biotechnology, EFB. It defines biotechnology as the integration of natural sciences and organisms, cells, parts thereof, molecular analogs for products and services. Now, let us see what are the further things that we study under biotechnology. Okay, these are some of the areas that biotechnology deals with. Microbe-mediated process. It involves making curd, bread, wine, etc. I mean, these are the things that you study in your +2 level topics. Okay, where microbes in human welfare is one chapter you'll be dealing with. Second one is in vitro fertilization. This is reproduction in human beings. Okay, assisted reproductive technologies, ART we call it as. Third one is synthesis and usage of gene, where molecular biology based techniques are involved. Fourth one is preparation of DNA vaccine. Vaccine is something that we give in order to prevent a disease. And last one is correction of a defective gene, where a gene is functioning in a wrong way, where we will be correcting it by giving it a right gene. So this is the areas where biotechnology deals with. These are the areas where we can have the applications of biotechnology into. Now let us see, as you know, there is in every field there will be principles, right? So in biotechnology, what are the principles that governs it? Let's see that. Now let us see the principles of biotechnology. What are the principles of biotechnology? There are two principles that is involved in biotechnological processes. One is genetic engineering. What is genetic engineering? The technique in which the genetic material, both DNA and RNA, either one will be in an organism, okay, DNA or RNA, is chemically altered and introduced into the host, thereby changing its phenotype. We take a DNA, okay, we change its chemical nature and we introduce in a host, thereby changing its phenotype. What do you mean by phenotype here? Phenotype means morphology, okay, and the host here refers to any organism where you are putting your gene into. The father of genetic engineering is Paul Berg. The father of genetic engineering is Paul Berg. Second principle is maintenance of sterile ambience. There is another name for this principle. We call it as bioprocess engineering as well. Bioprocess engineering. Now, in any technique, maintenance of sterile ambience is very essential. Why? As you can see here, it is necessary in chemical engineering for growing desired microbes or eukaryotic cell in large quantities for the manufacture of antibiotics, vaccines, enzymes, etc. So it is very important that you maintain a sterile environment so that when we are dealing with microorganisms, if you are putting out a media, there is a good chance of contamination where unwanted organisms will be growing, and this could tamper with your product which you are ultimately looking for it. So now let us see what are the three basic steps in genetically modifying an organism. Okay, let's see that. As you can see, the first step involves identification of DNA with desirable gene. This is the first and the most important step. You have to identify a gene. Okay, traditional hybridization technique lead to the inclusion and multiplication of undesirable gene and desirable gene. The genetic engineering helps to isolate and introduce only desirable gene. In this paragraph, you'll be reading here like traditional hybridization technique. These techniques are basically which you study in your strategies enhancement of food production, where we'll be dealing with plant hybridization traditional techniques. Okay, that technique they are meaning. That technique has a very good possibility of inclusion of as well as desired as well as undesirable gene. We need only the desirable gene. That particular point, genetic engineering helps us to locate desirable gene and help us in the way through. Second point, introduction of identified DNA into its host. Okay, a piece of DNA cannot multiply itself in the progeny cells of the organism. So it is integrated into the recipient genome. It multiplies and inherits along with its host DNA. Okay, so that means the identified DNA, which you have already asked, the identified DNA means the desired gene, which we discussed previously. That particular identified DNA is introduced into the host and it is multiplied along with the host. That's what it says. The maintenance of introduced DNA in the host and then transfer DNA to its progeny. That's his third step. That means you make sure your transferred DNA is confirmed in the host and it is getting transferred to its progeny as well. These are the three basic steps in genetic modifying an organism. So we have already seen three basic steps in genetically modifying an organism, right? Now, let us see the tools and the common terms that is used in these processes. We'll start with the first one. Gene transfer. Gene transfer, very simple term. Transfer gene from one organism to another. I'm not writing it here. This is a very common term. Second one, transgene. What is transgene? Now I have already told you gene transfer involves transfer gene from one organism to another organism. Transgene involves the gene that is getting transferred. The one gene that is getting transferred is called as a transgene. Third one, transgenic organism. Transgenic organism. An organism that possesses and expresses a transgene. That means from an organism you are transferring a gene. That particular gene is called as transgene. If an organism receives it, it expresses it, you call it as a transgenic organism. Remember the words possess and express is called as a transgenic organism. Fourth one, we call vector. Vector. What is vector? Vector is nothing but carrier of foreign gene. It is called carrier of foreign gene. Next, we are going to see what are the commonly used vectors. Commonly used vectors. I'm giving it as 5, okay? Commonly used vector. It is plasmid. You might have studied the definition of plasmid in your 11th standard topic, okay? What is plasmid? Circular extrachromosomal self-replicating DNA, okay? It is circular, double-stranded, self-replicating DNA. Now, next, we are going to see the term recombinant DNA. Recombinant DNA or rDNA. rDNA, recombinant DNA, the definition is nothing but native plasmid with a foreign gene. Okay, if you incorporate a foreign gene in a plasmid, we call it as rDNA. Seventh one, DNA ligase. Another name for this enzyme is molecular glue. Okay. More DNA ligase. It is used for joining the cut DNA fragments. Last one is restriction enzymes or molecular scissors. So these two terms you keep in mind. One is molecular glue, another one is molecular scissors. These are the most important 8 tools and terms that is used in biotechnology. So now let us see how foreign genes is introduced into a host cell. This is a picture that is in your textbook. As you can see, we have selected a foreign gene here and we